Good morning, good afternoon, or good evening, wherever you may be. We get a lot of people from other countries on these webinars, so I want to make sure I greet you appropriately. My name is Ann Brazo. I'm the CEO and founder of MPN Advocacy and Education International. Twice a year, we hear from some of our partners that have clinical trials that are bringing us up to date today about what's going on at each of their institutions and companies. And we do this because we feel that it is very important that our patients and our physicians, today is for patients and tomorrow will be for physicians who see MPN patients. And although not everybody may be on at the same time, the recordings are watched frequently by physicians that see MPN patients and by patients and caregivers. So we want to make sure we do this twice a year, even though at most of our webinars we do a 3-minute update. Today, we are allowing our guests to speak a little longer and show slides if they choose to. With me first is one of our partners from SOBI, Olivia Cholak, who's the medical science liaison. Olivia, are you with me? Where'd you go?
Yes, can you see and hear me?
Hello. So I'm going to let you take over from here and do your presentation. And if any questions come in, I may ask you, but we probably will just go through this, without questions till the end. Okay, thank you.
That sounds great. Okay,
Thanks
let me
for
just
being
kind of
with
share my
us today.
screen. Okay. One sec. All right, let me just get my presentation up. Okay. Can you see this? Okay. Okay, so thank you so much for having me, Anne, and everybody here. My name is Olivia Cholak. I'm a medical science liaison at SOBI in New York City, and I'm here to talk to you today about pacritinib, which I'm going to call Vonjo, our brand name, and it's used in thrombocytopenia and myelofibrosis, and of course, give you an update on our research efforts. Before I get started, just kind of a personal background on myself. Before joining SOBI, my background is in nursing, and I ran the nursing program for the MPN and the leukemia clinic at Mount Sinai. So I'm sure some of you here on the call have been patients of mine. If so, please reach out and say hi. I miss you guys. I think perhaps like many of you here on the call, before working in myelofibrosis, I'd never heard of it before. But working with patients and then learning from just the leading experts in the world like Dr. Mascarenhas, it really made me so passionate. I've worked on clinical trials, and it's so amazing, not just to see what goes on during drug development, but how when you have a patient who really doesn't have a lot of options left and see them respond really beautifully to a new therapy, it's just a really amazing experience to be a part of. So I just want to thank you, the patients, for everything that you do. And again, just thank you so much for having me here. So just to kind of give a brief background on SOBI and CTI, pacritinib Vonjo was developed by a company that was called CTI, which a year ago was acquired by SOBI. CTI was a very small company. Vonjo was the only drug that it made. SOBI is a global company that specializes in rare disease. So together, the philosophies of these two companies, they really complement each other quite well. So merged with CTI, SOBI is really becoming a leader in hematology, and Vonjo is a huge focus for the company. Now, when we're talking about Vonjo, what really sets it apart from other myelofibrosis therapies is that it works differently than the other JAK inhibitors. It's the only drug that's specifically for patients with severe thrombocytopenia, so platelets of less than 50,000. Now, before the approval of Vonjo, there really was a clinical unmet need in these severely thrombocytopenic patients. We know that other JAK inhibitors can lower the platelet count. So for somebody whose baseline platelets are already low, that might not be a safe option. And prior to Vonjo, none of the JAK inhibitors had been studied in patients who had platelets of less than 100,000. Now, you're here at this advocacy meeting. I know that this is a really well-informed group of patients. But for those of you who are new or just looking for some statistics, we know that myelofibrosis is a really rare disease affecting, a little less than 80,000 patients throughout the world. And as you may know, what's happening here is that, due to different mutations that are targeting the JAK-STAT pathway, we see scarring in the bone marrow. And then that's going to, dysregulate the normal production of blood cells. And that's why we're going to see anemia, low red blood cells, thrombocytopenia, low platelets in many of our patients. Now, about a third of patients with myelofibrosis have platelets of less than 50,000, and that's associated, we know, unfortunately, with worse outcomes and a median overall survival of 15 months. And like we've said, there's really a large unmet medical need for these patients. Up until this point, they've had just limited options. But here comes in Vonjo. And Vonjo is the first FDA-approved medication for severe thrombocytopenia and myelofibrosis. So, as I've said, patients that have these really low platelets, they're not candidates for most treatments because, like we've said, they could they could lower the platelets even further. And, this really presents, Vonjo presents an option for these patients to really show spleen and symptom improvement to just really benefit, their overall quality of life. So on a mechanistic level, what we're seeing here are the different cellular pathways that Vonjo inhibits. And like I mentioned earlier, these are the pathways that contribute to the disease and the symptoms that we see in myelofibrosis. So what's unique about Vonjo is it's the only agent, the only JAK inhibitor that spares JAK1. And that's why we think that patients that are on Vonjo don't see the same myelosuppression that we see on other JAK inhibitors. Like the other JAK inhibitors, it does inhibit JAK2, and that gives us that beautiful spleen and symptom response that we see. Another unique feature of Vonjo is that it inhibits this pathway IRAK1. IRAK1 is this pathway that ultimately downstream leads to these inflammatory cytokines, these chemicals that cause inflammation. So here's just another way that we can combat those symptoms of inflammation. And the fourth and final pathway that we're talking about is ACVR1. ACVR1 is implicated in iron storage in the liver. So by affecting the amount of iron available for red blood cell production, the thought is that Vonjo can help ameliorate anemia in patients as well. And here we get to Pacifica, which is the reason that we're here today to talk about our ongoing efforts. Here we just have a timeline of the events and studies that led to Vonjo approval. So Vonjo was approved in 2022. It got accelerated approval by the FDA based on patients with platelets of less than 50,000 and the spleen volume reduction that we saw. The approval is contingent on the completion of a confirmatory trial that's called Pacifica. Pacifica started in 2019. And after Vonjo was approved in 2022 and became available in the United States, now we have Pacifica only outside of countries beyond the United States. So this is a global study now. Here's the study design. And really the key points here, just to point out, are that this study looks at intermediate and high-risk patients that have severe thrombocytopenia. So those patients with platelets of less than 50,000. These are patients that have not been on a JAK inhibitor, or they've been on, less than 90 days of a JAK inhibitor. In total, it's going to look at 399 patients. We've enrolled just over 200. And patients are going to be randomized 2-to-1 either to pacritinib or a physician's choice medication. So that's going to be low-dose ruxolitinib, which is either 5 milligrams daily or twice daily, hydroxyurea, danazol, or corticosteroids. And the primary endpoints that we're looking at, like all the other JAK inhibitors, are the amount of patients that achieve a spleen volume reduction of at least 35% and a total symptom score reduction of at least 50%. And then our key secondary endpoints that we're looking at are overall survival, patient global impression of change, which is a subjective patient scale that really measures, how the patients feel, and safety. Here's another slide that's just going over those same endpoints that we just discussed. And then just to finish things out, here are our active clinical sites throughout the world. So you can see that we're currently on hold in Russia and Ukraine, but we've got a lot of other sites active. We're actively recruiting, and we're really excited just for the future of this study, what it'll continue to tell us about pacritinib use, about Vonjo use in these patients with severely low platelets. Just to finish things out, just including our indications and important safety information. But if you have any questions, please don't hesitate to reach out. We're always happy to talk to our patients. We always want to hear about your experiences. And again, thank you so much for having me. I think this is just a fantastic organization. I feel just really privileged to be a part of everything happening here today. And to the rest of the speakers, thanks for sharing everything that you've got as well. That's
Thank
it.
you
Thank
very
you.
much, Olivia. Thank you. That was really informative and I appreciate it. I have followed CTI since its inception, basically. And so I was very familiar with this drug and what it does and how different it is from the others. So, and I do remember you when you were at Mount Sinai, actually. So it's a pleasure to see you again. So
You
thank
too,
you very
Anne.
much and have a great day. And we'll move on to Silence Therapeutics and Curtis Rambaran, who's Dr. Curtis Rambaran, who's the Chief Medical Officer. Hi there, how are you?
Morning, I'm fine. Thank you very much for having us.
Yes, thank you. And I'm going to let you take over and do your presentation.
Okay, share my screen. I hope you can see. Let me just get this right. Okay.
I think the first thing you did worked, and now, you know, the link there. Yes, that's good.
Wonderful. Okay, great. So thanks again to everyone and the organizers and all the attendees. Delighted to be here today. I'm going to tell you a bit about Silence Therapeutics. So we are a global company. We are headquartered in London. We have our R&D and molecular design and scientists based in Berlin, in Germany, and a lot of the senior leadership currently in New Jersey, around 125 plus employees worldwide. And we chose the name Silence because we are really silencing our gene expression. We are silencing diseases using sRNA technology that is, very, very precise technology for inhibiting our gene expression. So Silence does have a long history in oligonucleotide development. And over that time, we have learned, different tricks as such to optimize our molecules. So currently, we have our sRNA molecule that's targeting a specific gene, in this case, for hematological disorders. We have a linker that then connects the sRNA molecule to a ligand. This is the GalNAc ligand. It's a naturally occurring sugar molecule. And basically what this ensures is that after injection, the drug, the sRNA, goes directly to the liver because the GalNAc ligand is bound to receptors in the liver. So it makes the molecule very targeted to the liver. And that's very important because we are really trying to specifically target the messenger RNA. So when we say silencing gene expression, we identify the abnormal genes causing diseases, and we sort of inhibit the messenger RNA, the sort of signals that result in a translation of that messenger RNA into abnormal protein. The good thing about it is that it is reversible. So this is unlike gene therapies that is not reversible and is once given and it's a lifelong treatment. Once you stop the sRNA, you can reverse any changes. We have a broad utility. We are targeting both rare and common genetic disorders. And we also have a range of programs in hematological disorders, cardiovascular, and also partnered programs with the big pharma. So our main asset, I should say our most advanced asset in development is for cardiovascular disease. This is an sRNA targeting elevated lipoprotein A. That's a protein that confers cardiovascular disease. And we have published data both in healthy volunteers and in patients cardiovascular disease patients showing very good efficacy. You'll see here that we have knocked down this protein or reduced this elevated lipoprotein A down to around 90% after single and multiple injections. So this is a very potent and a very targeted therapy. What I would like to talk to you about today is divesiran. That is our sRNA that's being developed for hematological disorders. So divesiran targets TMPRSS6 that modulates hepcidin. Hepcidin is your sort of master regulator of iron flux in the systemic circulation. So it decides how much gets to the gut, how much gets away, how much goes to other tissues. We have published positive preclinical data in animal studies showing a good benefit of the drug in a range of hematological disorders. And we did show proof of mechanism. So when we gave the drug to healthy volunteers, it was safe and it was very efficacious. So, healthy volunteers, we gave divesiran as a single injection, as a subcutaneous injection to 20 healthy volunteers. This caused a fourfold rise in hepcidin. That's the marker that we are trying to elevate and regulates iron, and it reduced your serum iron by 50%. So that really was very important because it showed that when we translated from the preclinical work into the healthy volunteers, the drug mechanistically works. Importantly, after a single injection, this effect persisted for at least 2 months. So potentially, this is the sort of therapy that you can give infrequent dosing. Equally important, we had no serious adverse events, and this really supported us going into a phase 1/2 study in polycythemia vera patients. We published this data at ASH in 2021. Now, we target in polycythemia vera because, as I'm sure many of you will know, this is the myeloproliferative neoplasm driven by exaggerated production of red blood cells and platelets, and the hallmark being elevated hematocrit, which is really an reflection of your total red blood cells. And the treatment goal is to get your hematocrit down to less than 45%, as per the guidelines, because we know that hematocrit above that does really increase your risk of cardiovascular events and thrombotic events. Because this is a rare disease, and the current standard of care, which includes a range of also chemotherapeutic agents, is still doesn't really suit all patients. Importantly, most of the polycythemia vera patients are iron deficient at diagnosis, and when they have repeated phlebotomy, this does exacerbate it. We did do some preclinical study in a model of polycythemia vera, and we showed that the drug does elevate hepcidin and have beneficial effects on red blood cells. So our hypothesis is that divesiran, or SLN124, previously called, will elevate hepcidin, regulating iron release to the circulation and restricting the supply to the bone marrow. And as we know, iron availability to the bone marrow is essential for red blood cell production. So potentially by restricting that availability, you could reduce the incidence and the hematocrit levels. We do have fast track designation, PV, and also orphan drug designation. So currently we have a phase 1/2 study in PV ongoing that's called Sanrico. We included adult patients with polycythemia vera. We are looking at safety, tolerability, and efficacy, and the pharmacokinetic effects of what the drug does in terms of systemic concentrations. The drug is administered, as I mentioned, by a subcutaneous injection. The phase 1 is ongoing. We are planning for the phase 2. The phase 1 is an open label dose finding phase. So both the practitioners and the investigators know what the patient is having. The primary outcome measure is to look at adverse events and the assessment of the number of phlebotomies over time. That's in the phase 1. So we really want to see if the safety from the healthy volunteer translates into patients, and to look at the effect on phlebotomies, which is crucial in these patients. For the phase 2, we are going to be looking at the number of patients who are phlebotomy-free after treatment. So just to summarize, divesiran has shown to be safe to date and has a potentially long duration of action, which hopefully should give additional options in the future to patients. We have shown that from a single dose, we can have good effects down to at least 2 months. We have designed a phase 1/2 study called Sanrico to identify the optimal dose for late development. We hypothesize and believe that patients will benefit from a reduction in phlebotomies and an increase in iron stores, which hopefully could translate to a reduction in fatigue. And when I say increase in iron stores, we are moving the iron away from the bone marrow, where it will increase the red blood cell count, and to other parts of the circulation and other tissues. Currently, it's a global study. We are actively recruiting outside of the US, and now we are open for recruitment in the US. So thanks very much for the opportunity to be able to very quickly give you a snapshot of our approach to polycythemia vera. If you want to find out more, we are listed on clinical trials, and also I've listed here a contact where we are very happy to take any questions at any time. We are really excited about this asset, and we really believe that it could give an alternative option in the future going forward for patients that may provide infrequent injections and be quite safe. So thanks very much.
Thank you very much. That was really interesting and unique. You know, it's a unique drug, and one that I believe our patients are going to need. So I'm hoping for the best, you know, and for continued education about what's happening. And this is exactly why we host these. So thank you so very much for that. And if you're a patient out there who's interested, anybody that mentions that, you know, that their trials are ramping up, you can take this information to your physicians, many of whom will be with us tomorrow. And we will be discussing these updates at that time with just physicians. Our next company and partner is Karyopharm Therapeutics, and Dr. Raman Garshut. He's the Senior Medical Director in Clinical Development. I hope I pronounced your last name correctly. I apologize if I didn't.
You did really well, Anne. Thank you, Anne, and
Thank
you know,
you.
the advocacy team. You know, real pleasure to be a part of this. So thanks for having us.
Thank you. And
Okay, give me a quick second just to share my screen. Okay, please let me know if you can see that as a full screen presentation.
I see that, but the language is to the side, so you might want to do a full screen.
Oh, I see. Display settings. Does that work?
Yes, that's good. Thank you.
Perfect, perfect. Okay. So hello, everyone. My name's Raman Garcha. I'm Senior Medical Director with Karyopharm Therapeutics. And, you know, what I'm planning to run through with everyone today is, you know, briefly touch on the unmet need with standard of care, describe our mechanism of action, and then to run through our phase 1/3 protocol and studies, now called Sentry, and then run through our phase 2 plan, now named Sentry 2. So, you know, excited to officially announce trial name change for Karyopharm in our myelofibrosis trials here. So this is a disclaimer slide. Okay, so who are we? Karyopharm is an innovation-driven pharmaceutical company, you know, using a first-in-class drug agent called selinexor, which targets nuclear export and related pathways with the aim of treating cancers and other major diseases, you know, focusing on unmet needs. Over 300 employees and 10+ years in research and development. And we're actually headquartered in Newton, Massachusetts, over on the East Coast. So what I'll be describing is, you know, Karyopharm's myelofibrosis trials. And as I mentioned before, our phase 1 to phase 3 study was formerly known as Export MF034. And we now have a nice name for the trial, and it's Sentry. So phase 1 has completed enrollment, and I'll go on to talk through some of that data. And we now have phase 3, which is a double-blind randomized trial, a combination of selinexor with ruxolitinib in JAK treatment-naive patients. We have that phase 3 now open for enrollment. We also have, as you can see on the screen there, an open-label phase 2 trial called Sentry-2, formerly known as Export MF-044. And I will go on to talk about the design of that trial, but that is also frontline JAK-naive patients with moderate thrombocytopenia, which is an important difference. So, please do take a look at the Karyopharm website, and the clinicaltrials.gov links are just at the bottom of that slide there as well. Okay, so the unmet need in myelofibrosis is well described, and generally speaking, touches on responses seen in standard of care, both SVR35 and TSS50, where it's been seen where less than 50% of patients are achieving this week 24 spleen volume reduction of 35% compared to baseline. Disease modification is obviously a hot topic and important to see how that matures in this setting. And JAKis may have a lack of evidence of disease modification with regards to this. Cytopenia, obviously, by virtue of the nature of the disease, is present, and in some instances the treatments can also be causing cytopenia as well. And obviously the disease in general, is associated with poor outcomes. It's associated with decreased survival and significant morbidity for these patients. And, that's really what we're trying to address here. So we have a slide here that describes the mechanism of action for selinexor. So as I said before, it's an XPO1 inhibitor, inhibiting the release of various cargo proteins from the nucleus. And, what you can see on the slide there is obviously the nucleus and the cell. And we have a color coding here. So, the top left is green, which is signifying JAK-STAT pathway inhibition. So with selinexor, we have evidence preclinically and in human cell lines that demonstrates JAK-STAT inhibition. But the color coding reflects the fact that, there are also other pathways that we may be acting on here with selinexor. So we have JAK-STAT in the top left, but in the blue boxes, you can also see how selinexor has been shown to have an effect on p53-driven cell death, cell cycle arrest, and NF-kappa B pathway inhibition, which may have an effect on cytokine production. So the references are found at the bottom of the slide there. This is a slide that, and again, I'm going to touch briefly on the main points of each slide because there's a lot of slides, and I just want to make sure I stay within our time limit here. But this is the broad plan for the Sentry trial, which again was formerly called Export MF-034. And it's the same protocol. It goes from phase 1, which is now completed enrollment, through to phase 3. So in the phase 1 portion, we had 24 patients, and it was obviously a dose escalation study. 2 dose levels were explored. So selinexor is dosed orally once weekly, QW. So two dose levels were explored, the 40 milligrams and the 60 milligram level. And 60 milligrams was found to be the recommended dose of selinexor in combination with ruxolitinib. So Sentry is a combination study, selinexor plus ruxolitinib. And, it had the primary endpoints you'd expect of safety. So just real brief, in terms of the makeup of the patients that were enrolled in the trial, there was a real spectrum of patients, which is important. It was quite representative of the MF population. Both the majority being primary myelofibrosis, and then pretty much equal proportions of secondary types of MF. Intermediate to risk was the biggest group in this phase 1 study. And again, equally proportioned high risk and intermediate 1 patients also. We also had a variety of driver mutations for our patients. And as you'd expect, it was predominantly JAK2 as the driving mutation we saw. So we have a slide here on safety. And again, there's a lot of information, so please do take a look in your own time. Hematological AEs that you'd expect to see. Nausea was controlled with prophylactic antiemetics. What we saw, which was really great, was a median weight gain throughout the cohort to 2.5 kilograms, with hemoglobin and platelet levels generally remaining stable from the start of study through week 24. So I just want to touch on the impressive efficacy that we saw on study. So the left-hand side of the screen is SVR35, and the right-hand side represents TSS50. So we actually saw early responses by week 12 for SVR35. And actually our week 24 SVR35 rate was 79%. So that was great for this patient group. And our week 24 TSS50 was a rate of 58%. Now you can see from the graphs below, especially the bottom left there, that in fact all patients in the efficacy evaluable population that were treated with 60 mg of selinexor achieved an SVR35 at any time. So this slide describes the durability of the data of the responses, and they were very durable. And that's important to know. For SVR and spleen volumes, we saw 100% probability of maintaining that spleen volume reduction with a median follow-up time of 32 weeks. The right-hand side of the slide, describes some of the suggestions towards disease modifications that I mentioned earlier. So we saw reduced cytokines measured at week 4 that were associated with some level of spleen volume reduction at week 24. We also saw variant allele frequency reductions in most of our evaluable patients. Okay, so on to our phase 3 trial, Sentry phase 3, which is now enrolling. So we're going to enroll approximately 306 patients. And again, this is a JAK-naive population, myelofibrosis population. It's a double-blinded trial with 2-to-1 randomization. And the 2 arms will be ruxolitinib BID, and the dose based off of the platelet count there, obviously, plus selinexor 60 milligrams POQW, versus the placebo arm, which is ruxolitinib BID plus placebo. So we actually have a co-primary endpoint for this phase 3 trial, and that would be SVR35 and TSS50 at week 24. The inclusion criteria you can see there on the screen, and is obviously available to look at on the ct.gov website as well. So I want to actually just finish up right now on phase 2 trial, Sentry 2. And this is a really interesting trial because we're going frontline monotherapy. So this is selinexor monotherapy upfront, JAK-naive patients in a group that has an unmet need, really. So this is a moderately thrombocytopenic population. And the reason we were pointed towards this is in the Sentry trial, phase 1 that I just described, when we had patients which were dosed at a very low dose of ruxolitinib in combination with selinexor, we still saw multiple SVR35 responses. So even when 5 mg BD or less of ruxolitinib was used long-term in combination with selinexor, we still saw responses. So that was quite encouraging for us to proceed to this phase 2 trial. And you can see the plan there. So it's a 2-arm open label study. And again, moderately thrombocytopenic population. So platelets 50 to less than 100 for inclusion on this trial. And, you know, there's 2 arms, 60 milligram arm and a 40 mg arm. And what's also interesting about this trial is if patients are not achieving the splenic volume responses by weeks 12 or week 24, we have an option to add on JAKis, either ruxolitinib, pacritinib, or momelotinib, depending on which region the patient is in. So, you know, that's real interesting because we're starting with Seli mono, and, you know, the expectation is the patient would continue on monotherapy. However, if they're not reaching various SVR requirements, the investigators will have an option to add on these optional JAKis. So I'm going to stop there. I've been talking for a while, and, you know, thanks for your attention. And, you know, what I ran through there was the mechanism of action for selinexor, the Sentry and the Sentry-2 plans, and some of the data. So thanks so much for your time.
Oh, it's fascinating. And I'm so encouraged. You used the word encouraged, and we are as well, and our patients should be as well. And it's just nice to know all of this work is being done, and so thoughtfully, to be honest. I mean, it's just really been nice. And so thank you again, and I'm sure we'll see you tomorrow as well for the physician presentation.
For sure. Thanks so much, Anne.
Thank you. Have a good day. And we'll move on to Protagonist Therapeutics, who is another partner with us, and Dr. Efede Ajari, who's the Executive Director of Clinical Development. Is he with us?
Yes, I'm here.
Okay.
Hello, everybody. Thank you for having us here. I'll very quickly start to share my screen.
Looks good.
Okay. Thank you very much for your time. Thank you for creating this avenue for us to have a discussion with you about Protagonist and the interesting things that we are doing. For those that don't know, Protagonist is a biopharmaceutical company located in the South San Francisco Bay Area. Our primary platform is based on our proprietary peptide technology with rusfertide, or PTG300, as our lead investigational drug candidate. It's a synthetic mimetic of the naturally occurring hormone hepcidin. And as you all know, hepcidin helps to regulate iron metabolism and controls the absorption, storage, and distribution of iron in the body. Very, very quickly, I just want to create some foundational statements around the disease and how we are approaching it. PV is a type of blood cancer that affects the bone marrow where blood cells are made. So in PV, the body makes too many red cells, and we have an elevated hematocrit, which is the percentage by volume of red cells in the blood as a hallmark of the disease. So technically, it might be thicker than normal, thereby limiting the natural flow of blood, and this could increase the risk of unwanted blood clots. And we have some long-term diseases that could be associated with any increase in thrombosis that affects the heart and other organs. It can happen at any age, but it's commoner in people aged 50 to 70 years, with a median survival of about 20 years. Our treatment goal is always to keep hematocrit at less than 45 percentage, and also to minimize complications. It's again suspected due to the elevated hemoglobin and the hematocrit measurements. Our diagnosis, you have a number of patients presenting with other diseases like hypertension, elevated enzymes. About a third of even have enlarged spleens, itching, and a few other things. Now, there are other symptoms, on a day-to-day basis, like fatigue, early satiety, bone pain, sexual dysfunction, excessive sweating, and other things that even worsen over the course of the disease. And in thinking about our program, we brought everything into view. The current way of treating right now is, to regularly remove the blood to reduce the number of red cells in the blood, a procedure just like donating blood. It has its own clear limitations, bringing about fluctuations in hematocrits, even as we try to maintain the consistent blood level controls. It's always very difficult. But for such a procedure, it takes up to 4 hours. There could be a very massive impact in the quality of life of the people that are experiencing it as a disease, and its potential to interrupt work and the overall productivity in life. It's something we are very much cognizant of. Now, in addition to phlebotomy that we just described, aspirin, hydroxyurea, interferons, as well as ruxolitinib, are some of the treatment options and approaches, for PV that we have. And there are a number of ongoing interventional phase 3 studies in PV, from the use of interferon to including low-dose aspirin or hydroxyurea, even to combining a direct oral anticoagulant with low-dose aspirin. They are all being used. But our approach is a little different, because for the disease called PV, there's what you call the hyperactive erythropoiesis that brings about an accumulation of an excessive amount of red cells. Now, there is the human hormone called hepcidin, which is central to the regulation of iron that the body needs for red cell production. Now, by binding to the export protein, as we see on our screen illustrated, ferroportin, iron is redistributed away from the hemoglobin compartment and into storage sites. There is where we are basing the scientific basis of our own drug, and we correctly tagged it a hepcidin mimetic. Now, rusfertide targets this imbalance of iron metabolism by targeting the ferroportin receptor in this manner, similar to hepcidin, and gets in the way of delivering iron to plasma. So through its effect in helping to restrict iron availability for erythropoiesis, we are currently using rusfertide for conditions characterized by excessive erythropoiesis, such as PV. We very recently got into a partnership agreement with Takeda to jointly develop rusfertide, and we are still investigating rusfertide in ongoing phase 2 and 3 clinical trials for PV. There's the ongoing verified phase 3 pivotal study designed for about 250 patients. Enrollment was just finished for the verified study just a few weeks ago. Now, there is a revived phase 2 study for 70 participants with an additional 3-year open label extension part. The core study part has been completed and the open label extension is ongoing. But there is also the long-term extension for those patients that will complete revive to roll over into a long-term study for us to continue to study hepcidin, and for the patients hopefully to continue to benefit from what we are seeing with rusfertide. Now, there's a Pacific phase 2 open label single arm design with a dose escalation part by subject cohort. We already completed that, and it's also relevant before we get into the next set of slides to mention that rusfertide has orphan drug designation and fast track status for PV as we speak. Okay, I really want to quickly demonstrate some of the results that we have seen from the REVIVE clinical proof of concept study, where we added on rusfertide to standard of care. Now, in this study, all the patients, were initially phlebotomized. We wanted to keep their hematocrit levels to less than 45%. And we wanted to ensure that as we give doses of rusfertide subcutaneously, we were able to see whether or not we would maintain hematocrit at less than 45%. So we performed analysis of response. We performed studies where we looked at whether or not we actually reduced phlebotomies and utilized a validated myelofibrosis symptom assessment form as a tool to see, if the PV and the symptoms were responding to our treatment. And I will just quickly show you some of the highlights of our results. Now, we are looking at the second part of the REVIVE study, the part 2, which is the blinded randomized withdrawal part of the study. Responders are defined as subjects who did not receive any phlebotomies and they maintained hematocrit control without becoming phlebotomy eligible. And they were able to complete 12 weeks of treatment. As you see here, 69.2% of these patients, which is 18 out of 26, responded to rusfertide in our full analysis population. So if you exclude those that even discontinued early and did not complete this part, but if you even include them, you see that 60% of patients, 18 out of 30, responded to rusfertide in this randomized population, clearly indicating that rusfertide met our primary efficacy endpoint for response. This study also showed that rusfertide was able to provide sustained and durable control of hematocrit, with treatment resulting in consistent maintenance of hematocrit at less than 45% through 2.5 years, and this is still ongoing. Then this is also just to show, referring to the patient-reported outcomes we mentioned earlier, across the different domains, we saw very remarkable improvements across the board. This was objectively assessed, and we're very impressed at the kinds of responses we got from the patients when they use those scales. Now, based on the REVIVE study, we also reported very good long-term safety profile for rusfertide. The most common treatment emergent adverse events were the injection site reactions, which were localized. They were mild to moderate in severity, and they really generally decreased in incidence over time. They were all deemed to be histaminic in nature, and the use of any over-the-counter antihistamines were found to be helpful in these cases. Overall, about 77% of these treatment emergent adverse events were moderate at most. About 21% were grade 3, and there were no life-threatening or fatal treatment emergent adverse events reported. So let us also note here that the mean duration of exposure was about 105 weeks, which ranged from 3 weeks to 182 weeks, and this continues to increase. So still talking about safety, overall, about 14 patients, which was about 20% of the 70 patients we studied, experienced serious adverse events. There were 3 cases of basal cell carcinomas. There was one case each of a number of conditions, from atrial fibrillation to AML and squamous cell carcinoma, which were seen in placebo patients. Of course, we still needed to report them and study them very closely. But the nature of the SAEs observed were consistent with the kinds of diseases that are anticipated in the disease population. And most of these SAEs were assessed by the investigators as unrelated to rusfertide. So to enable us chart a course forward, to potential registration, we are capitalizing on the results of the phase 2 REVIVE study using rusfertide. So we are designing, or we have designed, a phase 3 study in patients. It's in theory a 2-part study, but technically a 3-part study, because Part 1 has A and B parts. So the key inclusion criteria to enroll patients 18 years and above that meets the revised revised 2016 WHO criteria for diagnosis of PV. Now, these are patients that require at least 3 phlebotomies due to inadequate hematocrit control in 28 weeks before randomization, or at least 5 phlebotomies within 1 year prior to randomization. Once enrolled, all these patients are initially randomized into Part 1A, which is the randomized double-blind placebo-controlled period for 32 weeks. During this time, the enrolled patients are randomized 1-to-1 in a blinded fashion and given rusfertide or placebo added onto their ongoing treatments for PV. And randomization has been stratified by their ongoing treatment for PV. The Part 2 is the open label part where all subjects, that completed Part 1A successfully would receive rusfertide on top of whatever standard of care that they have for 20 weeks, going up until week 52. Then the Part 2, which is the third part, is the long-term extension for 104 weeks, where they will all continue to receive rusfertide. And the key endpoints are just the proportion of those patients that are achieving response, defined as the absence of phlebotomy during the second half of the double-blind portion, and also the mean number of phlebotomies within the double-blind treatment period. So in summary, we have very compelling data to really show that there's value that is being demonstrated using rusfertide in our PV patients that, before joining our study, they required frequent phlebotomies with or without any, background cytoreductive therapy. So we're still studying them. We've been looking at them for way over 2.5 years, and we are really seeing very rapid, sustained, and durable hematocrit control, as well as well-documented positive improvements in symptom scores. We will really continue to see what we would learn from the phase 3 verified study to confirm the efficacy as well as the safety of rusfertide. We've actually completed the enrollment in the last quarter. We've enrolled approximately 250 patients, and we will continue to learn the very exciting things we are learning about rusfertide. I will stop here, but I really want to thank you all for offering us within Protagonist the opportunity to present our data.
Thank you so very much. We really appreciate it, Dr. Ojara. That was very thorough and very interesting. Thank you. So we are going to move on now to Merck and Company Incorporated and Dr. Uzor Agbu, who is the Senior Clinical Director. Hello, Uzor. How are you?
I'm doing well, Anne. How are you?
I'm good. Thanks for joining us today.
All right, let me share my screen. Sorry, give me 1 second.
Looks good.
Okay. And I will, as a word of warning, I have had Zoom restart on me 2 times during this call so far. I hope it won't happen during this presentation, but I will be back within a minute if it does. So as Anne pointed out, I'm a senior clinical director at Merck, working on bomedemstat, which is our LSD1 inhibitor, which we see as having applications across a number of the myeloproliferative neoplasms. It is an inhibitor of LSD1, which is an epigenetic enzyme that is important for the maturation and differentiation of blood cells. Looking here, the reason we think, or we believe LSD1 will have an important impact in the myeloproliferative space is based on its activity. It has an upstream activity where it affects the mutant hematopoietic stem cells that go on to produce all the, either its platelets or red blood cells that lead to all the issues we see across ET, PV, and also MF. And it also has a downstream impact. Here we're showing it for ET, where the activated megakaryocytes lead to an increase in inflammatory cytokines. There's the elevated platelets that I mentioned earlier, and an increase in growth factors that eventually leads to the bone marrow fibrosis and the splenomegaly. Inhibition of LSD1 affects all of these things. It reduces the inflammatory cytokines, which will reduce the constitutional symptoms patients experience. It reduces the number of platelets, which reduces the risk of thrombosis and bleeding. And it reduces the growth factors, which leads to reduced deposition of reticulin and collagen, and hence leading to a decrease in bone marrow fibrosis. But importantly, and this is what would apply across a number of the different MPNs, it also reduces the malignant cell population. And by doing that, in this case, we know JAK3, MPL, CALR, all their canonical mutations, and they lead to, depending on the patient, could lead to ET, could lead to PV, and could lead to MF. This inhibits that malignant stem cell population, regardless of if it's JAK2, CALR, or MPL. That's why we believe it has applications across. I will present data primarily here for ET, which you have seen before, and show you what our next steps are. And also talk a little bit about PV. We do not have results for that yet, but I will talk a little about what that trial looks like. This is, as I said, it's data you've seen before. It's the phase 2 trial in ET, where we treated 73 patients. They had a median age of about 67, a median time since diagnosis of about 83. And they were all types. They had JAK2, CALR, MPL accounting for about 90%, which is what you would expect to see for ET patients. And we also had 9% of triple negative patients that don't have any of the 3 canonical mutations. You see there's a wide range for the MPN SAF, their symptom score. The median was 16, but it ranged from 0 to 70 or 74. And as you would expect, most of these patients are coming off hydroxyurea, since this was a relapse refractory trial. 70% had their immediate prior therapy as hydroxyurea. But you do see we had some patients with anagrelide, interferon, busulfan, ruxolitinib, and also a combination of anagrelide and hydroxyurea. A quick summary of what we saw. So at week 24 for these ET patients, 95% of them had a treatment response, which is defined as a reduction in platelet below 400,000. 28% of those of the 28 patients who 28 patients who reached 48 weeks had a durable response, which again is a reduction below 400,000 for a minimum of 12 weeks. There was also a reduction in the variant allele frequency that was regardless of what mutation the patient had between CALR, JAK2, and MPL. 67% of patients had a decrease in their variant allele frequency. Another important part of it is there are patients who were homozygous for mutations. We saw initial inklings that some of those patients are converting from being homozygous to heterozygous, which you would expect, but being homozygous makes your disease more severe. Being heterozygous should make it slightly less severe. That's something we're continuing to evaluate in our future trials, and we think that may have an impact and additional aspect of disease modification that this agent might bring. Importantly, for the ET patients, there was no reduction in their hemoglobin across their duration of treatment. This is important for ET. It's a very different situation for PV patients, but I will take you back to where the mechanism of action, where it affects the mutated stem cells. If those mutated stem cells are producing excessive platelets, that's where we're seeing the activity. If they're producing excessive red blood cells, we would expect that would then affect the red blood cells and still have an impact on platelets, but less of an impact on platelets, and we wouldn't see that maintenance of a hemoglobin level. Going on to patient symptoms, we did see significant improvement in symptoms. Here we see of the 12 participants that had the worst symptoms, there was 75% of them noticed an increase in those symptoms overall after a 48-week treatment period. Based on the MPN SAF scale, the average improvement with bone pain was about 0.4 and fatigue was about 1.1. From a safety, we feel it's safe and tolerable. As you're aware, one of the issues we've seen with this agent is dysgeusia. They have been relatively mild and time limited. We are continuing to evaluate that because as we're conducting trials across the different MPNs, we're noticing quite a difference in the rate of dysgeusia. This seems to be at the highest rate, and we're wondering, is there an interaction between with bomedemstat within ET patients? But there'll be more forthcoming on that. So far, the dysgeusia, as I said, has been mild, low grade, and manageable with dose alterations or changes, timing of when you have a dose. And the dysgeusia is unique in that it leads to an accentuation of taste. It doesn't make things taste bad per se, but it leads to an accentuation of taste. So sweet things tend to taste sweeter. Obviously, that may be an issue for some patients, but like I said, it has been mild, generally time limited, and manageable with alterations and lifestyle modifications. It was based off the strength of those results that we saw in ET patients in that phase 2 trial that we late last year launched a phase 3 trial looking at patients, ET patients who have an inadequate response or they're intolerant to hydroxyurea. Our hope is to enroll about 300 patients who are randomized to either bomedemstat once daily or to the best available therapy, which could include anagrelide, busulfan, interferon, or ruxolitinib. Patients will be treated for approximately 52 weeks before we have a primary analysis. But at that point, patients are able to go on to an extended treatment phase and either switch from their best available therapy or move to bomedemstat based on if the study reads out positive. Like I said, this study opened up at the end of last year. The primary endpoint for the study is a composite endpoint where we're looking at a durable clinical hematologic response, which would mean a reduction in the platelet count below 400,000 for a minimum of 24 weeks, which is longer than what we typically see. The NCCN guidelines tend to talk about a 12-week duration, but we're aiming for a 24-week reduction of platelets continuously over that period. And concurrent with if they have elevated white blood cell count, that blood cell count should come below 10, and they shouldn't have any major bleeds or thromboembolic events during this entire 52-week period. So a number of different things combined in there, but we think that should demonstrate what the impact is of bomedemstat for ET patients. As secondary endpoints, we're also looking at using the MF-SAF to look at the total symptom score and what impact we have there. And we, as I said earlier, we're continuing to evaluate in terms of disease modification. What does it mean? Are we, what extent are we reducing the VAF, and what impact can that potentially have on patients? And also looking at what impact do we have on bone marrow fibrosis. When we spoke last year, I had talked about our PV study in which we were seeking to enroll 20 patients. That study is now fully enrolled, and we're following up those patients for the specified 30-week time point to look at an endpoint of can these patients maintain a hematocrit below 45 for a minimum of at least 12 weeks in the absence, continuous 12 weeks in the absence of phlebotomy during this 36-week period. Again, this is a phase 2. This is the first time we are testing bomedemstat among a pure PV population. Based on these results, we will evaluate and see what would be the next step. And once again, patients who do complete their 36 weeks have the option to continue therapy on bomedemstat. As a last slide, sort of an overview of where we are and how we're expanding our MPN program here at Merck. I talked about PV and ET. There is a myelofibrosis trial. We had a phase 2 trial, which has been completed. There is an ongoing collaboration. This completed one was bomedemstat as a monotherapy. There is an ongoing collaboration to look at bomedemstat in combination with ruxolitinib, which was presented at ASH last year. That's still ongoing, and we're evaluating the data to decide what are the next steps for bomedemstat, either as a monotherapy or in combination with ruxolitinib or potentially any other JAK inhibitor. I've walked you through what we're doing with essential thrombocythemia. We have our first phase 3 launched now. We expect more things to come in the near future, but for now we have that one phase 3 in patients who are refractory or have an inadequate response to hydroxyurea. The PV trial I just mentioned, which is ongoing, is fully enrolled. We expect that we should have results and initial analysis of that by the end of this year. The one thing I have not mentioned is our bomedemstat extension study, which we expect for all of these patients across our phase 2 or phase 3 program, given that most of these trials run for approximately 1 year, they are able to roll over and get on the extension study if they are willing, in which we'll be able to look at what is a longer term impact. 1 year is a limited amount of time to look at what disease modifying type of endpoints. Going on to the extension study will allow us to continue monitoring them, and we expect that extension study to go on for a number of years so we can truly measure what is the potential impact of Bomedemstat across the different MPNs that we have.
Fantastic. Thank you so very much. It was excellent. And we really appreciate all of your updates, but this was really great. And I know I'll see some of you tomorrow for our updates for our physicians. And so in closing, I hope this was very informative to our patients who joined us today. But in closing, I want to just send out a reminder to you patients who are already using the 4 drugs, one of the 4 drugs that are available to you. That would be Jakafi, Ojjaara, Vonjo, or Besremi. And if you need help getting your medicine, for Jakafi, you can go to insightcares.com. For Ojjaara, you can go to togetherwithgskoncology.com. For Vonjo, ctiaccess.com. And for Besremi, pharmacentiasource.com. So if you need help getting your medicine, please reach out to those folks. And we will see the physicians and many of you who spoke today tomorrow. And have a great day. This was so informative and so wonderful. Thank you so much.